All students think that the linkage and recombination is very complicated topic, but if you are observe carefully, that is easy only. But before listening about the video which is related to linkage and recombination, it is better to complete the listening and reading of monohybrid cross and dihybrid cross. If you are understood complete details of monohybrid cross and dihybrid cross and the laws of Mendel, then only you will come here and see about the linkage and recombination. Without understanding of Mendel monohybrid cross, dihybrid cross and the laws of Mendel, you can't understand the linkage and the recombination. There is the links in between the Mendel work and linkage and recombination. Hence, once go through my videos, I have uploaded the videos which are related to monohybrid cross, dihybrid cross and then laws of Mendel. After watching that, come here and see about the linkage and recombination. Okay, let's start about the linkage and recombination. Linkage and recombination. What is the linkage? What is the recombination? First we will discuss what is the linkage and what is the recombination and types of linkages. Then after that we will apply this linkage and recombination to experiments. First, linkage. Linkage. Linkage means physical association of genes on same chromosome. Physical association of genes. Physical association of genes. Physical association of genes on chromosome is called a linkage. Here, if you see, this is the chromosome. One minute, I will the chromosome large. This is the chromosome, gene A, gene B. There is a two genes which are present on the chromosome. Next, we can see homologous chromosomes in human body and homologous chromosomes in diploid organisms. Same another chromosome also present. Okay, these are the genes which are present on same chromosome. How many genes are there? Two, A and B. Here linkage is physical association of genes on the chromosome. In linkage, A and B genes, if the A and B genes are linked genes, they are avoid to dissociate, they are avoid to segregate, they are avoid to separate in next generation. According to Mendel, what happened? Inheritance of one gene is not depend on inheritance of another gene, but in linkage, both genes as it is transfer into the next generation. There is no exchange of genes because of linkage. Hence, the gene A and gene B avoid the segregation or separation. They are as it is transfer into the next generation. The gene A and gene B, they are avoid to segregation and they are as it is transferred to the next generation. See, if you are understand recombination, then you can understand about linkage very well. I will explain recombination here. Recombination. Recombination means Recombination means exchange of genetic material, exchange of DNA or exchange of genetic material in between non-sister chromatids of homologous chromosomes. Homologous chromosomes is called recombination. Exchange of genetic material in between non-sister chromatids of homologous chromosomes is called recombination. First, you should understand what are non-sister chromatids and what are the homologous chromosomes. If you observe in human body, each chromosome is occurs in a pairs. Chromosome number 1 is occurs in a pair, means 2 chromosomes are there, same chromosomes which contain same genes at same place or same loci. Hence we can say that human beings contain 23 pairs of chromosome. First pair is similar each together, second pair is similar to each together, third pair is similar to each together, means they contain same genes at same place, but they may show variation in alleles, forms of alleles. One chromosome may contain dominant allele, another chromosome may contain recessive, but genes are same. That alleles are belongs to same genes. See here diagram. This is one chromosome, and this one is one chromosome. Okay, if you observe the structure of chromosome, each arm, this is one arm, this is one arm, this is one arm, this is one arm. Each arm of chromosome is called chromatid, not chromatin, that is chromatid. Chromatin is different. Chromatin is present inside the nucleus in all normal cells which are not involved in cell division. The nucleosomes of cell combine together and form the chromatin, that we can discuss in next chapter. Here chromatid means arm of a chromosome. All chromatids attached by a structure that is called centromere. Centromere is used for the attachment of chromatids of chromosome. Next, what are the non-sister chromatids? If you give number to the chromatids 1, 2, okay, next here also 1, 2, these 2 and 2, these 2 are sister chromatids. They contain same genes at same loci. See here A only, their gene A is present at same place. Hence these 2 are sister chromatids, and chromatid 2 and chromatid 1 are non-sister chromatids. These are non-sister chromatids. It may contain the gene B, okay, but 1 and 1, these 2 are sister chromatids. BB is present in both. That is the difference between sister chromatids and non-sister chromatids. Here 2 and 1 are non-sister chromatids. What is the recombination? Exchange of DNA between non-sister chromatids of homologous chromosomes. What are the homologous chromosomes? These 2 are homologous chromosomes. Why? Because they contain similar type of genes at same place. This is one pair, chromosome number 1 pair or chromosome number 2 pair, chromosome number 3 pair. Already you know that in diploid organisms chromosomes are occurs in a pair. Each pair is called homologous chromosomes. Here these two are homologous chromosomes. In homologous chromosomes also these two are non-sister chromatids. There is a exchange of genetic material in between these two chromosomes during the meiosis. Mainly at prophase 1 of meiosis. Simply you can say that during the formation of gametes, there is a exchange of genetic material in between the two non-sister chromatids of homologous chromosomes. This leads to variation in sexually reproducing organisms. This will show the variations in sexually reproducing organisms. There is a variations we can see in between the parents and offsprings. That variations are because of this recombination. See, now observe after recombination. Observe the chromosomes after recombination. There is a exchange of genetic material in between the non-sister chromatids of homologous chromosomes. See here, the small part of chromosome of chromatid 2 is enter into the chromatid 1, and the small segment of DNA or chromosome of chromatid 1 enter into chromatid 2. That is the exchange of genes, exchange of genetic material in between the non-sister chromatids of homologous chromosomes. That will show the variation. That will show the variation. Okay, that is about the recombination. If you come to here, if genes are very closely linked, very close together, that is called linkage. They avoid separation. They avoid separation during the formation of gametes. If crossing over, see, this is A, I will show by dots. This is A and this is B. Gene A, gene B. They are very close together. During recombination, A and B both transfer into the one offspring only. Why? Because there is no chance to separate A and B during recombination. Okay, in case recombination exactly occurs here, then only they will separate. Exactly it will occur at middle in between A and B, then only A part enter into another chromosome. Otherwise recombination if it occur here, there is no separation of the genes. If recombination occurs here, no separation of genes. If recombination occur here, no separation of genes. That is called linkage. That is called linkage. Physical association of genes in which the linked genes as it is transfer from parents to one offspring. Okay, parents to offspring. They will not separate. Okay, example here A and B are not separate. Linkage again divided into two types, that is incomplete linkage and complete linkage. Incomplete linkage and complete linkage. Incomplete linkage, in incomplete linkage what happen? Genes are arranged very close to each together. See here, incomplete linkage A, B. Two genes are very close to each other. Okay, they avoid the recombination. Then complete linkage. If you observe the complete linkage, there is a little bit distance. Distance is there in between the complete linkage. 1 minute. Okay, linkage again divided into two types. Linkage again divided into two types, that is complete linkage, incomplete linkage. Complete linkage means the genes, linked genes are tightly arranged, or linked genes are very close together. In complete linkage, we can see less recombination. They avoid the separate, hence less recombination. Incomplete linkage means there is some distance is there in between the A and B. If recombination occurs here, there is a chances to separation of genes. That is called incomplete linkage. In complete linkage, linked genes are arranged tightly, close together. In incomplete linkage, linked genes has some distance in between that. In incomplete linkage, there is a chances to recombination. Compared to incomplete linkage and complete linkage, in complete linkage we can see less recombination. In incomplete linkage, we can see more recombination. When distance between two genes is very less, recombination also less. When distance between two genes is more and recombination also more. That is the difference between complete linkage and incomplete linkage. Now what we have to discuss? We have to apply this incomplete linkage, complete linkage and recombination to experiments. There is one scientist, his name is T.H. Morgan. He did experiments on Drosophila and he identified that there are genes, those are called linked genes. They avoid the dissociation, they avoid the separation, and few genes are show complete linkage, few genes are show incomplete linkage. And Morgan also proved that if linkage is more, in complete linkage less recombination is formed, less recombinants are formed. In incomplete linkage, more recombinants are formed. Example for complete linkage. Example for complete linkage, that is color, eye color and body color are controlled by the two genes, eye color and body color. Body color is controlled by the alleles Y and Y plus. Eye color is controlled by the genes W and W plus. W plus is dominant allele, W is a recessive allele. Y plus is dominant allele and Y is a recessive allele. Here what happened? The W and Y genes are the genes which are responsible for eye color and body color are tightly linked in the Drosophila. It is identified by the T.H. Morgan. Next, incomplete linkage. Incomplete linkage. Incomplete linkage example, eye color and wing size. Eye color and wing size. Eye color and wing size are example for the incomplete linkage. Okay, in NCERT they given the diagrams also which prove that if the more the linkage, if the more tightly packed the genes, the less recombination. If the less the linkage means incomplete linkage, more the recombination. Okay, that diagrams we have to discuss now. before going to see about diagrams, we will discuss few points that are related to Morgan experiments on Drosophila for linkage and recombination. Okay. T.H. Morgan is one scientist. He selected the animal Drosophila. Drosophila, also called as fruit fly, for his experiments to observe about the linkage and recombination. Why he selected Drosophila? Because it has a short lifespan, then we can grow it easily in laboratory in the glass jars, and they will show the contrasting characters. Because of all those features, T.H. Morgan selected the Drosophila for his experiments. T.H. Morgan, also called as father of experimental genetics, father of experimental genetics. Why? Because he did lot of experiments on Drosophila for identification of linkage recombination. He also proposed the term criss-cross inheritance. If you observe the Drosophila, there is a few genes in Drosophila, they are compactly packed, they are tightly packed, or they show the complete linkage. Few genes in Drosophila incompletely linked. Here T.H. Morgan did dihybrid experiments in Drosophila. Which experiments? Dihybrid cross. Dihybrid cross. T.H. Morgan did dihybrid crosses in Drosophila, but he got different result than the Mendel. Mendel did dihybrid cross in the pea plants, but he got 9:3:3:1 ratio of phenotypic characters in F2 generation. But the result of T.H. Morgan is different in Drosophila compared to the Mendel dihybrid cross result. That is, after that he identified that in dihybrid cross of Drosophila we are getting different result because the genes are present on same chromosomes. The genes are linked genes. Few genes of Drosophila are linked genes. But in Mendel experiments, genes are present on different chromosomes. Hence, inheritance of one gene not affect on inheritance of another gene. During formation of gametes, definitely they will be separate if they are present on different chromosomes. But here genes are present on same chromosomes. Hence, they avoid the separation or segregation. Because of linkage in dihybrid cross of Drosophila, Morgan got different result than the Mendel result. And apart from that, few genes which are present, few linked genes which are present on X chromosome, means sex linked genes, sex linked genes, sex linked linkage, that also leads to formation of different result in the dihybrid cross of Mendel. And after the dihybrid cross of after the dihybrid cross of Morgan did dihybrid crosses in the Drosophila. Finally, he identified that there is a genes, okay, there is a genes which are responsible for the body color, and eye color. The genes which are responsible for the body color and eye color of Drosophila will show the complete linkage, because the body color responsible gene and eye color responsible gene is very close together. Hence they are show the complete linkage. If you observe the alleles of body color that are small y, this is recessive allele, and small y plus is a dominant allele. Already we know that in Mendel monohybrid cross, dominant allele will give its phenotypic character. The body color, dominant body color in Drosophila is brown color, means Y plus allele will give brown color to the Drosophila. Then recessive body color is yellow. Yellow is recessive body color in Drosophila. Then if you observe the phenotypes, if both small y's are there, then only we can see the formation of yellow color body. Why? Because small y is recessive. Allele which is responsible for body color. When small y express its phenotypic character, or when recessive allele express its phenotypic character in homozygous condition only, means if small y is present, which will give yellow color only. Next, small y plus small y plus and y, there is a dominant allele. Y plus is a dominant allele, and when dominant allele is there, then we can see the formation of dominant character only. Why? Because dominant allele can express its phenotypic character in both homozygous condition and heterozygous condition. Hence, here we can see the formation of a brown color body. Brown color is a dominant color, then the yellow color. Then both y plus are there in this condition also, we can see the brown color body. Then if you see the eye color, the recessive allele for eye color is W. W is recessive for the eye color. Next, W is a recessive allele. W plus is a dominant allele. For the eye color. What is the dominant eye color? Dominant eye color is red. If red color eyes are there in Drosophila, that is the dominant. Next, recessive eye color is white. White color eye is recessive. Eye color is white. If you observe the genotypes, small w, both recessive alleles are there, which will give the white color eye. Then W plus and W is there, which will give red color eye. Why? Because this W plus allele is a dominant allele. In heterozygous condition also, we can see the expression of phenotypic character. When both W plus are there, which leads to formation of red color. Why? Because dominant allele always express its character, its phenotypic character in both homozygous condition and heterozygous condition. In NCERT they given this diagram. What is the diagram? The cross between two Drosophilas which will differ in body color and eye color. Here two characters he taken to cross. Hence this is also one type of dihybrid cross. This body color and eye color in Drosophila will show the complete linkage. Means the genes which are responsible for body color and eye color, certain body color and certain eye color, they are not changed. Mainly the Y Y means recessive body